All right, so all reactions are going to require an input of energy to get started. Some of them might be a lot of energy, some of them might be a little bit of energy. What we would call this is activation energy, so the energy required to start a reaction. So here's the reactants. You have to add a little bit of energy. So if we look at the x-axis versus the y-axis, the free energy used, in this case, we're dealing with an exergonic reaction, so you're adding a little bit of energy to get the reaction started, and then once you actually get it underway, boom, you get this drop in free energy. You're releasing energy into the environment, and you're creating those products through the course of the reaction, and you've lost energy. But it took a little bit of energy to get started. Kind of like you could say glycolysis. You add 2 ATP, but the reaction eventually yields you 2 additional ATP, or 4 ATP in total, which nets you 2 ATP. So what enzymes do is they catalyze or lower the activation energy, E subscript A, so energy activation. So activation energy, again, is the energy required to start a reaction. So if we look here, this blue line resembles a reaction that is uncatalyzed. And now if we have some kind of enzyme that catalyzes this reaction, what the enzyme might do is it will lower the energy required to start the reaction. So they bring down the activation energy in order to speed reactions. So let's look at, what an enzyme could hypothetically do. They're catalysts that get reactions started quickly, and enzymes are typically catalysts. So we have this disaccharide here, maltose, and we have an enzyme that is going to be, it's three-dimensional. If you don't remember from previous biology courses, its three-dimensional shape is what's going to determine its specificity. Typically, your enzymes are going to be very specific for one type of reaction and no other types of reactions. And so in this case, we have an enzyme that's perfectly catered to break down maltose into 2 monosaccharides. So maltose happens to be glucose bound to glucose. It's a disaccharide. And what happens is it will bind to an active site in an enzyme, and then that enzyme will stress the molecule in a specific way to catalyze the reaction, or the spontaneous breakage of the bond between the 2 glucoses, yielding 2 monosaccharides, or 2 glucose molecules. So this enzyme is specific for breaking down maltose. So some things they can do to lower that activation energy is, for example, bringing the substrate into proximity with another, or it could strain, in this case, it can strain that bond in a way that causes that bond to break. And it could do so chemically, or it may do so chemically. Okay, and then an example, or a term that I would use to describe this reaction would be hydrolysis, without even looking at the formula. You're breaking something down. This is likely to be a hydrolysis reaction. Okay, so again, back on enzymes, let's talk about the anatomy of an enzyme. Most of them are going to be composed of proteins, and those proteins, those enzymes, are going to have an active site which binds to substrate, or the reactant, the thing that's going into the reaction. And again, like I said, your enzymes are going to have a high degree of specificity, which is determined by its three-dimensional shape. So only certain substrates will fit. And an enzyme is only going to catalyze a single type of reaction, and it's going to be determined by the enzyme's 3D shape. So here you have an enzyme that's very specific in its active site. And what happens is you take substrates together. In this case, this enzyme is specific for fusing 2 things together. So it has a specific location where one substrate will bind, and has another specific location where another substrate will bind, and it might stress them in a way that causes them to form covalent bonds between one another. And so you could say, like, this is fructose and this is glucose. This is fructose and this is glucose, and they bind to the enzyme, and what it will do is it'll fuse them together. We could say this is the disaccharide sucrose, for example. It fuses them together through a dehydration synthesis reaction. All right, so enzyme substrate complex is what they form when those substrates bind. So here you have this green guy. It's not specific for the active site, but these 2 are, this red and orange, or red and yellow ones are, they bind perfectly to make that, disaccharide. And when they do that, when they bind to the active site, we call this the enzyme substrate complex. So at this moment in time, you're forming the enzyme substrate complex, and then eventually the enzyme will release that product, and the product will float away. So the enzyme is bound to the substrate in the enzyme substrate complex, which will be important when we get to inhibitors. So the result is that the product is formed and the enzyme is unchanged at the end of the reaction, meaning it didn't permanently change its conformational shape. It can now be reused for more reactions or recycled. So enzymes are going to speed up reactions by orienting substances for maximum reactions, inducing strain on the substrates like I mentioned earlier to maybe break bonds, or even strain in a way that causes them to form new covalent bonds that they otherwise wouldn't in low energetic conditions. Also adding charge to substrates by adding chemical groups temporarily. So the rate of reaction is going to be dependent on the concentration of things. Also, the rate of reaction is going to be dependent on the conditions that the enzyme is sitting in. Most enzymes have evolved to have a very specific sweet spot for temperature as well as pH. And so the temperature and pH of the solution or the surroundings of that enzyme might actually impact the rate of reaction. But for the most part, if you're using the ideal conditions already, the rate of reaction of an enzyme is going to be measured by how fast an enzyme can convert a substrate to a product. Substrate concentration is going to increase the rate of the enzyme until all of its active sites are saturated. That just means that you have a beaker full of enzyme and you throw in a little bit of substrate, and it's going to convert that substrate into the product at a certain rate. And you could keep adding more and more higher concentration of substrate. Eventually, you're going to oversaturate those enzymes to where the rate limiting step is actually the concentration of enzyme. And so then you would need to add more enzymes, kind of like the cereal and the milk. You can keep eating the cereal, whatever. I don't need the cereal and the milk. All right. So enzyme shape is going to determine its specificity and efficiency. Some enzymes require other molecules to complete the active site. Okay, so we have three different terms that we need to know here. Inorganic cofactors. So inorganic, we already know that it's not going to have carbon and hydrogen both. It's not going to have both carbon and hydrogen involved. It's an inorganic, typically an ion or a mineral that can bind to the enzyme. An example of this is magnesium, which actually binds to the enzyme. Did I say enzyme? A mineral magnesium, which binds to the enzyme DNA polymerase, referring to it as being an enzyme, polymer. It's making a polymer of something. What is it making a polymer of? DNA. So just by reading this word, or you could say this term, you could probably infer that this is an enzyme that is going to make a polymer of DNA. Magnesium is important as an inorganic cofactor for that. It will bind to that enzyme, activating it before it becomes active, before it becomes an active enzyme. Another term we need to know is prosthetic groups. So those are going to be non-protein organic or inorganic molecules or elements that permanently bind to an enzyme. So some vitamins are going to do this. So an example of an inorganic prosthetic group would be something like iron, for example, which binds to a heme protein. And the heme protein, which is bound to iron, is used to create the full hemoglobin molecule, the full hemoglobin protein. And we know that that protein pretty much acts as a transporter of oxygen. And the iron is actually what activates the heme groups. So that would be an example of a prosthetic group. So lastly, we have organic cofactors or coenzymes, and we know they're going to contain both carbon and hydrogen because they're organic. They're going to be non-protein organic molecules that temporarily bind to enzymes in order to activate them. An example of this is vitamin B7 or biotin, which is actually required for the enzyme pyruvate carboxylase, an enzyme that converts pyruvate to oxaloacetate. Another example of this would be vitamin C. Vitamin C actually binds to the enzyme that synthesizes collagen. All right, so factors affecting enzyme rate, like I alluded to earlier, you're going to have temperature and pH be the main contributors or environmental contributors to the activity of an enzyme. So like we mentioned earlier, substrate concentration is another factor. You can only get so high in substrate concentration before the active sites on the enzyme become oversaturated and it's at max capacity. So the other two factors are temperature and pH. So optimal temperature and optimal pH will vary depending on the environment, and extreme temperature and pH will typically denature the enzyme. When you denature an enzyme, you're breaking apart hydrogen bonds, covalent bonds, things that are going to contribute to the three-dimensional shape of the enzyme. And if you remember, the enzyme's function is dependent on its three-dimensional shape. If you're breaking down its three-dimensional shape, it's going to reduce its effectiveness. So it changes its shape, so it will not function. So we could say that, let's see, the temperature is the x-axis and the rate of reaction is the y-axis. And this blue line, you could say, at 4 degrees Celsius could be an enzyme that evolved to have prime reactions or prime activity at very low temperatures. So this could be like maybe an archaea living in Antarctica, an enzyme that is produced by a certain archaea. And then we have 37 degrees Celsius. We could say that that's an enzyme of some kind of bacteria that lives in our gut. And then this green line here could be an enzyme that's produced by an archaea that lives in a hot spring in Yellowstone. So all of the enzymes that organisms will produce will typically function ideally at specific pH and temperature. All right, so besides the concentration of substrate and besides the temperature and besides the pH, there's other factors that can affect enzyme rate. And again, we're kind of talking about, not again because I don't think I mentioned it, but we're talking about a numbers game. Typically in a solution, you're going to have many enzymes, not just one enzyme. So enzyme rate, we're not talking about the rate that a singular enzyme will conduct the conversion of substrate to product. We're talking about how fast a collection of enzymes in a solution of a certain concentration of substrate at temperature and certain pH will create product. So this is kind of a numbers game. So factors that can affect the enzyme rate is a chemical that may interfere with the enzyme's activity. So things we might have are inhibitors. So inhibitors can affect enzyme activity. They're going to inhibit enzyme activity. There's a few different ways we can inhibit enzyme activity. So inhibitors may work in a variety of ways. First, we have competitive inhibitors. Competitive inhibitors are going to bind to the active site of an enzyme, blocking substrate binding. So normally an enzyme, here's this purple thing is an enzyme. This little shape here is the active site. The substrate is perfectly formed to fit in the enzyme's active site is perfectly formed to fit the substrate into its active site. And so this is the way a normal enzyme would function. However, if you have a competitive inhibitor included in the mix, it might have a similar shape to the substrate, allowing it to bind to the active site of the enzyme. And what happens is when the competitive inhibitor binds to the active site, the substrate will therefore bounce off of the active site or that partial active site, causing it to no longer bind. And therefore, this enzyme is temporarily inhibited for as long as that competitive inhibitor is bound to. So the competitive inhibitor is actually competing directly with the substrate. Next, we have non-competitive inhibitors. So your non-competitive inhibitors are not going to compete with the binding of the substrate. They're actually going to bind to something, or they're going to bind to a location we would call an allosteric site. So here's an allosteric site, a site that is not the active site within the enzyme, that's going to cause it to change in conformation. So this enzyme, originally shaped like this, which would normally bind the substrate perfectly, well, when you have a non-competitive inhibitor binding to that allosteric site, what happens is the substrate can no longer bind to the active site, so it just changes the three-dimensional shape of the enzyme. So competitive inhibitors compete with substrate binding. Non-competitive inhibitors bind to an allosteric site, changing the conformation of the active site of the enzyme, therefore inhibiting substrate binding. Now, lastly, at least when it comes to types of inhibitors, is we have uncompetitive inhibitors. So uncompetitive inhibitors, what they're going to do is they're going to bind to the enzyme substrate complex, like I mentioned earlier. So when the substrate binds to the enzyme's active site, you form temporarily this enzyme substrate complex. So what it's going to do is sometimes they're actually going to be irreversible, meaning they're forming permanent covalent bonds. Sometimes they're going to be reversible, but regardless, what makes uncompetitive inhibitors distinct is that they bind to the substrate enzyme complex. So normal enzyme active site is shaped like this. Green guy likes to bind to that and create the product. Cool. Competitive inhibitors are going to be shaped similarly to the substrate, and they're going to bind to the active site competing with substrate binding, preventing the enzyme's activity. And then you have the non-competitive inhibitors, which bind to an allosteric site. And so when they bind to that allosteric site, it might change the conformational shape of the enzyme. Therefore, when substrates attempt to bind, they might bounce off, or they're not going to fulfill the reaction of the enzyme. Now, lastly, we have uncompetitive inhibitors, and what they do is they lock that substrate into place within the enzyme. And so let's say you have thousands of enzymes in solution. Well, let's say some of those enzymes bind to some substrate, and those enzymes bound to the substrate are locked in place by this uncompetitive inhibitor. They lock that substrate into place. Therefore, not allowing the enzyme to create the product, and two, it cannot serve as a catalyst for a new reaction. It cannot be recycled immediately because it's stuck, bound to substrate temporarily by an uncompetitive inhibitor. So to give an example of an uncompetitive inhibitor is finasteride, otherwise known as Rogaine. We know that that prevents or could help treat male pattern baldness. The way we typically get male pattern baldness is an abundance of dihydrotestosterone. And the enzyme that converts testosterone into dihydrotestosterone is 5-alpha reductase. So what finasteride does is it actually binds to the enzyme substrate complex of 5-alpha reductase, temporarily preventing it from moving from one substrate to another substrate to make more product. So it, in a sense, reduces the production of dihydrotestosterone. So allosteric enzymes can be activated or inactivated. Allosteric enzymes are just enzymes with allosteric sites. They have sites that are not the active site that can be bound to by inhibitors or activators. Typically, our non-competitive inhibitors will bind to the inactive or bind to that allosteric site, causing it to become inactive. So in this case, we have allosteric inhibition. Here's our enzyme. The allosteric inhibitor or the non-competitive inhibitor will bind to that allosteric site, causing a conformational shift. It distorts, in this case, it uses the term distorts its active site, disallowing the substrate to bind. Now we also have other enzymes that won't even become active until an activator is bound to the allosteric site. So we have the active site, which typically won't bind to the substrate, and if you have that allosteric activator that binds to it, okay, it changes the three-dimensional shape in a way that allows substrate to bind. So the takeaway from this is that some enzymes will have an allosteric site, which can be acted upon by inhibitors, or some of them might be capable of being activated on by activators. So let's talk about a neat concept called feedback inhibition. So let's say your cell needs to make some kind of end product, some product that's very useful for the cell, but you don't want too much of it. You want just enough end product. You want just enough of this product, this square, this yellow square. Well, your first reaction, in order to get to this, you're going to have to undergo many enzymatic processes to convert the molecule into another molecule and into another molecule and into another molecule, eventually until you get this useful thing at the end. But again, you don't want too much of it. Your enzyme 1 is going to take this green triangle, that's its substrate. It's going to convert that green triangle substrate into a product, and that product is a purple rectangle. Now your enzyme 2, the next step in this assembly line, is it's going to take up that product of enzyme 1 and it's going to convert it into a blue blob. So enzyme 2 converts the purple triangle into the blue blob. And then enzyme 3 is going to take that blue blob, and this is the final step to create this end product. It takes that blue blob and it makes that yellow triangle, or triangle, square. It's going to make the yellow square. And now again, like I mentioned, your cell wants to make just enough of that end product. It doesn't want to make too much of that end product, especially if these enzymatic processes take ATP. You don't want to use too much ATP to make too much end product that maybe you might not end up using. You want to make just enough. Sometimes this end product could have an affinity for an allosteric site on an enzyme. This could be the first enzyme, the second enzyme, even the third enzyme. What that allosteric or what that end product might do is inhibit one of these enzymes from producing substrate or producing, product using substrate. In this case, it is non-competitively inhibiting the first enzyme. So the green triangle can't bind to the active site, and now you're not producing any more of this purple rectangle. Therefore, you have no substrate for enzyme 2. Therefore, you're not creating this third substrate or second substrate. Therefore, you're not creating any end product. So it could put a temporary halt on this assembly line in case you don't want to make too much of a certain product. All right, question. When an enzyme catalyzes a reaction, what is it doing? A, it lowers the activation energy of the reaction. That's actually the right answer. Let's look at the other ones, though. So B, it raises the activation energy. Nope, it's lowering the activation energy. C, it acts as a reactant. No, it's catalyzing the reaction. The reactant is going to be the substance going into the reaction. And then D, it is used once and then discarded. Typically, enzymes are not used once and discarded. Typically, enzymes can be recycled. Okay, so this looks like a complicated process. I just wanted to use this as an example of a signal cascade, or you could say a baton passing from protein to protein that would eventually lead to change within the cell. So insulin receptor kinase. So kinase, we already know it's going to phosphorylate stuff. Insulin receptor, we know that's going to bind to insulin. And typically your receptors are going to be made out of proteins that are embedded in the cell membrane. So here you have the insulin receptor. It looks like insulin is these little pink balls. Insulin binds to this protein. And it looks like this is probably the insulin receptor kinase. So when the insulin receptor binds to insulin, or when insulin binds to the insulin receptor, the activated receptor phosphorylates something because it's a kinase. Kinases phosphorylate things. So it phosphorylates this protein. We don't need to know the name of this, but IRS1 protein. So IRS1 becomes phosphorylated. Then what happens is IRS1 bumps into something else. It's going to activate PI3 kinase. Again, you don't need to know any of the jargon necessarily. Just kind of try to conceptualize the signal cascade. So that IRS1 is going to bump into PI3 kinase, which then catalyzes the addition of a phosphate group to the membrane lipid PIP2, thereby converting it to PIP3. So your activated PI3 kinase, remember this was activated by IRS1, kinase is going to add phosphates to things. It adds a phosphate to PIP2, making it PIP3. Then PIP3 can bind to another protein kinase, something that's going to phosphorylate things. You're kind of like passing a phosphate from one group to another group to another group to another group. And so this PIP3 is going to phosphorylate, or it's going to bind to protein kinase called AKT, which is activated by other protein kinases. Now from here, you get another signal cascade. You get a continuation of the signal cascade. So this AKT is going to catalyze the phosphorylation of proteins, leading to an increase in glycogen synthase. So glycogen synthase activity. Glycogen. If you remember glycogen from the macromolecules, it's the way that our cells, or animal cells, will store sugars. Glycogen synthase is probably an enzyme, enzymes, probably going to synthesize something. What is it synthesizing? Glycogen. Glycogen synthase is going to increase in activity. So as we know, insulin, when it binds to our cells, it allows us to take up sugars into the cells. Now when it takes up sugars into the cells, what our cells will like to do, especially if we have an excess of sugar, is store it as glycogen. Now, how do we get that glucose into the cell? Well, what happens is this AKT is also signaling a cascade in order to increase the production and you could say the recruitment of glucose transporter, GLUT4, to the membrane. So this is just kind of a very short, very simplified depiction of how insulin binding to a receptor eventually leads to a protein signaling for the creation or the recruitment of transporters to get glucose into the cell. And then in addition to that, creating more of the enzyme that's going to help store sugar.